The impossible spark, life of Earth, Abiogenesis. The story of life on Earth is the grandest epic imaginable, a saga spanning billions of years, driven by cosmic forces and atomic ballet. But before the dinosaurs roamed, before the first fish swam, before even the simplest bacteria divided, there was nothing alive. For hundreds of millions of years, our planet was a swirling maelstrom of rock, water, and gas, utterly devoid of the animating spark we call life. How, then, did the inert become the living? How did the inanimate atoms of carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur self-organize into the breathtaking complexity of a cell, a redwood tree, a whale, or a human being? This is the profound question at the heart of abiogenesis, the scientific inquiry into how life arose from non-living matter. It is a question that has tantalized thinkers for millennia, from ancient philosophers pondering spontaneous generation to modern biochemists meticulously recreating primordial conditions in laboratories. The journey from a lifeless planet to one teeming with biodiversity is not a single miraculous leap, but a series of incremental, chemically plausible steps. Each step, though seemingly small, represents a monumental hurdle overcome by the sheer forces of thermodynamics and the relentless march of chemical reactions. Understanding abiogenesis is not merely an academic exercise. It touches upon our deepest questions about existence, our place in the universe, and the very definition of what it means to be alive. To begin, we must dispel a common misconception. Abiogenesis is not evolution. Evolution describes how life changes and diversifies once it already exists. Abiogenesis describes the preceding foundational event, the emergence of that very first life. Nor is abiogenesis the same as the origin of the universe, cosmology, or the formation of Earth, geology. While these events set the stage, abiogenesis focuses squarely on the chemical and physical processes that cross the boundary from non-life to life. The concept of spontaneous generation once dominated thought, suggesting that living organisms could simply materialize from decaying matter, maggots from meat, mice from grain. This notion, championed by Aristotle and widely accepted for centuries, was finally laid to rest by the elegant experiments of Francesco Redi in the 17th century and, definitively, by Louis Pasteur in the 19th century. Pasteur's swan-necked flask experiments demonstrated unequivocally that life only arises from pre-existing life, a principle known as biogenesis. However, while Pasteur disproved spontaneous generation for existing complex life forms, he didn't address the fundamental question of how the very first life came to be. This left a colossal void in our understanding, a gap that modern science has been striving to fill. The modern scientific understanding of abiogenesis began to take shape in the 1920s with the independent proposals of Alexander Oparin in Russia and J.B.S. Haldane in Britain. Both scientists, building on Darwinian principles and nascent chemical knowledge, hypothesized that the early Earth's conditions were vastly different from today's, and critically, that these unique conditions would have favored the spontaneous formation of organic molecules. They envisioned a, quote, primordial soup, oceans brimming with increasingly complex organic compounds, energized by lightning and UV radiation, gradually coalescing into self-replicating entities. This Oparin-Haldane hypothesis became the bedrock for subsequent research. Crucial to their hypothesis was the idea of a reducing atmosphere. Today's atmosphere is oxidizing, rich in free oxygen, O2. While essential for us, oxygen is highly reactive and would quickly break down complex organic molecules. Oparin and Haldane argued that the early Earth's atmosphere would have lacked free oxygen. instead being dominated by gases like methane, CH4, ammonia, NH3, water vapor, H2O, and hydrogen, H2, with significant amounts of carbon dioxide, CO2, and nitrogen, N2. In such a reducing environment, organic molecules could form and persist without being immediately destroyed. The Oprah and Haldane hypothesis remained theoretical until a young graduate student named Stanley Miller, working under the guidance of Harold Urey at the University of Chicago, decided to test it experimentally. In 1952, Miller designed an ingenious apparatus. He created a closed system circulating water to simulate the ocean, methane, ammonia, and hydrogen to simulate the early atmosphere. He then introduced an energy source, electrical sparks to mimic lightning. Below, he included a condenser to cool the gases, allowing water and any dissolved compounds to collect, simulating rainfall and the pooling of chemicals in the oceans. After just 1 week of continuous operation, Miller analyzed the contents of his primordial soup. The results were astonishing. He found a variety of organic molecules, most notably several amino acids, the fundamental building blocks of proteins. He also detected sugars, lipids, and other precursor molecules. The Miller-Urey experiment, as it came to be known, provided the first powerful empirical evidence that the basic organic building blocks of life could spontaneously form under plausible early Earth conditions. It was a landmark achievement, igniting the field of abiogenesis research and demonstrating that the leap from inorganic to organic was not impossible, but rather a predictable outcome of fundamental chemistry. While subsequent research has refined our understanding of the early Earth's atmospheric composition, perhaps less hydrogen-rich than Miller assumed, with more CO2 and N2 variations of the Miller-Urey experiment using different gas mixtures, have continued to produce a wide array of organic molecules. Furthermore, we've discovered other plausible sources for these early organic compounds. Meteorites, particularly carbonaceous chondrites, have been found to contain a diverse collection of amino acids, nucleic acid bases, sugars, and fatty acids. This suggests that a significant influx of complex organic molecules could have been delivered to Earth from space, augmenting whatever formed in situ on the planet. The formation of simple organic monomers, like amino acids and nucleotides, was the crucial first step. But life is not just a collection of monomers. It's a hierarchy of ever-increasing complexity. The next major hurdle was polymerization. How did these simple monomers link together to form complex polymers like proteins from amino acids and nucleic acids from nucleotides? In living cells today, this process is catalyzed by sophisticated enzymes and intricate cellular machinery. On the early Earth, such machinery didn't exist. Researchers have explored several mechanisms for abiotic polymerization. One promising avenue involves dry land surfaces, particularly those subject to cycles of wetting and drying, such as tidal pools or volcanic fumaroles. When water evaporates, it concentrates dissolved monomers, making it more likely for them to react and form bonds. Clay minerals, with their layered structures and charged surfaces, have also been shown to act as catalysts, providing templates and concentrating sites for polymerization. For instance, montmorillonite clay can facilitate the formation of RNA strands from activated nucleotides. Hydrothermal vents, deep beneath the ocean's surface, present another compelling environment. These volcanic fissures spew superheated, chemically rich water, creating steep temperature and chemical gradients. Such environments are rich in metal sulfides, which can act as catalysts for various organic reactions, including the formation of peptide bonds between amino acids. The vent hypothesis suggests that life may have originated not in a sunlit primordial soup on the surface, but in the dark, energetic depths of the ocean. These environments are protected from harsh UV radiation and asteroid impacts, offering a more stable cradle for nascent life. So we have a planet with a reducing atmosphere, abundant energy sources, and mechanisms for forming simple organic molecules, and even polymerizing them into more complex structures like short proteins and nucleic acids. This brings us to the core mystery. What truly defines life? And what was the minimal set of features that the first living system possessed? At its heart, life is defined by a few key properties. Self-replication, the ability to make copies of itself, passing on its information. Metabolism, the ability to acquire and utilize energy from the environment to maintain its structure and perform its functions. Compartmentalization, the ability to maintain an internal environment distinct from the external environment, typically within a membrane. Evolution, the ability for its information to change over time, leading to adaptation and diversification. Today's life integrates these functions seamlessly. DNA stores genetic information. Proteins catalyze metabolic reactions and build cellular structures, and lipid membranes define the cell's boundaries. This intricate interplay presents a chicken or egg problem. Which came first? DNA needs proteins to replicate and express its information, and proteins are encoded by DNA. This apparent paradox led to one of the most significant theoretical breakthroughs in abiogenesis research, the RNA world hypothesis. The RNA world hypothesis proposes that early life was based primarily on RNA, not DNA or proteins. RNA, or ribonucleic acid, is a versatile molecule. Like DNA, it can store genetic information. Crucially, unlike DNA, some RNA molecules can also act as enzymes, catalyzing chemical reactions. These catalytic RNAs are called ribozymes. This dual capability makes RNA a compelling candidate for the first self-replicating, metabolically active molecule. In an RNA world, RNA molecules could both store the instructions for their own replication and catalyze the very reactions needed to build more RNA. Over time, errors in replication would introduce variation, allowing for natural selection to favor more stable, efficient, or accurate self-replicating ribozymes. This would be the dawn of evolution at a molecular level. Experimental evidence has provided strong support for the RNA world hypothesis. Researchers have synthesized ribozymes in the lab that can self-replicate short RNA strands, demonstrating the fundamental feasibility of this idea. Furthermore, within our own cells today, relics of an RNA world persist. Ribosomes, the cellular machinery responsible for protein synthesis, are themselves ribozymes. They use RNA to catalyze the formation of peptide bonds. This suggests that RNA played a much more central catalytic role in ancestral life before proteins took over most enzymatic functions, and DNA became the primary information storage molecule due to its greater stability. However, the RNA world hypothesis still faces challenges. How did the very first RNA nucleotides form abiotically? The synthesis of nucleotides, particularly the sugar ribose, is chemically complex. Research by John Sutherland and his team has shown that, under plausible early Earth conditions, a complex series of reactions involving simple carbon compounds could lead to the simultaneous formation of ribose and the nucleotide bases, integrating what were previously thought to be separate, difficult steps. This prebiotic plausible synthesis offers a powerful counter-argument to the critiques of the RNA world. Even with self-replicating RNA, life still needs a boundary. The ability to maintain an internal environment distinct from the external one is a hallmark of all living cells. Without a membrane, the precious organic molecules and catalysts would simply diffuse away. and the chemical reactions of life would be impossible to control or concentrate. This is where compartmentalization comes in. Lipids, or fatty acids, are relatively simple organic molecules that can spontaneously self-assemble into vesicles or micelles in water. These structures are essentially tiny bubbles with a double-layered membrane, remarkably similar to the cell membranes of living organisms. These protocells could have encapsulated early self-replicating RNA molecules and other organic compounds, concentrating them and allowing for localized chemical reactions. Crucially, these protocells can also grow by incorporating more lipids and then divide, much like simple cells. This allows for a form of protocellular selection, where vesicles that successfully enclose and protect their contents might have a survival advantage. So the grand narrative of abiogenesis begins to unfold. Abiotic synthesis of monomers, simple inorganic molecules energized by lightning, UV radiation, and volcanic activity, form organic monomers like amino acids, nucleotides, sugars, and fatty acids. These may have formed on Earth or been delivered by meteorites. Polymerization. These monomers link together to form polymers, short proteins, peptides, and nucleic acid strands, RNA. This could have occurred on mineral surfaces, in evaporating pools, or near hydrothermal vents. The RNA world, RNA molecules emerge as the central players, capable of both storing genetic information and catalyzing their own replication and other metabolic reactions. This marks the beginning of molecular evolution. Compartmentalization. Protocells. Lipids self-assemble into vesicles, encapsulating these self-replicating RNA systems, creating protocells that maintain a distinct internal environment. Emergence of DNA and proteins. Over time, DNA, being more stable, takes over the role of primary genetic information storage. Proteins, with their vast catalytic versatility, gradually assume most enzymatic roles, relegating RNA to intermediary and regulatory functions. This transition marks the last universal common ancestor, LUCA, the point from which all known life on Earth descended. This detailed, multi-step process, rather than a single event, highlights the intricate interplay of chemistry, physics, and geology over hundreds of millions of years. It suggests that life wasn't an accident, but an almost inevitable outcome, given the right conditions and sufficient time. Let's delve deeper into each of these stages, exploring the scientific evidence, the remaining questions, and the exciting frontiers of research. Stage 1. The primordial soup and its ingredients. While the Miller-Urey experiment demonstrated the synthesis of amino acids, the precise composition of the early Earth's atmosphere remains a subject of ongoing debate. Geochemical evidence suggests that the atmosphere might have been less strongly reducing than Miller initially assumed. Instead of being dominated by methane and ammonia, it might have been more neutral, with higher levels of carbon dioxide and nitrogen, similar to volcanic emissions today. Even in these less reducing atmospheres, however, experiments have shown that organic molecules can still form, albeit sometimes in lower yields or requiring different energy sources, like UV radiation combined with metal catalysts. Beyond atmospheric synthesis, other sources for organic molecules are crucial. Hydrothermal vents, particularly alkaline vents, are now considered prime candidates. These vents release hydrogen-rich, warm, alkaline fluids into the cooler, acidic, iron-rich early ocean. The chemical disequilibrium across the vent's porous mineral structures, often iron sulfide like McKenna-White, creates natural electrochemical gradients, similar to those that power metabolism in modern cells. These gradients could have driven the synthesis of organic molecules, even from CO2, providing both energy and building blocks. Some scientists even propose that these vents could have been the very birthplace of metabolism, preceding the formation of complex informational molecules. Another significant source, as mentioned, is extraterrestrial delivery. Carbonaceous chondrite meteorites, remnants from the early solar system, are known to contain a surprising diversity of organic compounds, including over 80 different amino acids, some of which are not found in Earth-based life: nucleic acid bases, adenine, guanine, cytosine, thymine, and uracil, and even sugars. During the Hadean Eon, the earliest period of Earth's history, from its formation to about 4 billion years ago, Earth experienced intense bombardment by asteroids and comets. These impacts would have delivered vast quantities of organic material, effectively seeding the planet with ready-made building blocks. While some of these would have been destroyed by the impact, a significant portion could have survived, especially if delivered to water. The concept of a primordial soup also requires careful consideration. Was it a global ocean-spanning broth, or were organic molecules concentrated in specific locales? The consensus leans towards localized concentrations. Tidal pools, evaporating lagoons, the pores within hydrothermal vents, or even the surface of clay minerals could have acted as concentration devices, bringing dilute monomers close enough together to react. Concentration is key because many of the reactions required to build life are unfavorable in dilute solutions. Stage 2. Polymerization, from monomers to macromolecules. The leap from simple amino acids and nucleotides to complex proteins and nucleic acids is a chemical Everest. In modern biology, this process is exquisitely controlled by ribosomes and polymerases, enzymes that precisely link monomers together using energy derived from ATP. On the early Earth, these sophisticated mechanisms were absent. One of the most widely studied abiotic polymerization mechanisms involves dehydration reactions. The formation of a peptide bond between two amino acids, or a phosphodiester bond between two nucleotides, releases a molecule of water. In aqueous environments, water is abundant, making these reactions thermodynamically unfavorable unless energy is added or water is removed. This is where the idea of wet-dry cycles becomes compelling. Imagine a tide pool or a volcanic hot spring that periodically evaporates. As water recedes, the concentration of monomers increases dramatically, pushing the equilibrium towards polymerization. When the pool refills, the newly formed polymers can be rehydrated and potentially undergo further reactions. Experiments have shown that repeated cycles of wetting and drying on mineral surfaces can indeed lead to the formation of polypeptides and oligonucleotides of significant length. Mineral surfaces play a critical role here. Clay minerals, such as montmorillonite, have highly ordered crystalline structures and charged surfaces. These surfaces can absorb and concentrate organic molecules, orienting them in specific ways that facilitate bond formation. Furthermore, some metal ions, like zinc or nickel, embedded in clays can act as catalysts, lowering the activation energy for polymerization reactions. Think of them as rudimentary enzymes, providing a template and a reactive environment. For RNA, clay minerals have been shown to catalyze the formation of strands up to 50 nucleotides long, which is a significant achievement in abiotic synthesis. Hydrothermal vents offer another unique environment for polymerization. The steep temperature gradients mean that hot, concentrated solutions of monomers could be quickly cooled, leading to precipitation or self-assembly. The presence of metal sulfides, particularly iron-nickel sulfides, within the vent structures can also catalyze peptide bond formation. These minerals possess catalytic properties remarkably similar to some of the active sites found in modern enzymes, suggesting a deep evolutionary lineage. A critical aspect of polymerization is chirality. Most biological molecules exhibit homochirality. They exist in only one of two mirror image forms, like your left and right hand. Amino acids in proteins are almost exclusively L-isomers, while sugars in DNA and RNA are D-isomers. Abiotic synthesis typically produces a racemic mixture, an equal blend of both L and D forms. How did life select for only one form? This remains an active area of research. Possible mechanisms include chiral mineral surfaces acting as selective catalysts, circularly polarized light from nebulae, or even chance events amplified by selection. Some theories suggest that early life might have tolerated a mix, with homochirality emerging later as a refinement for greater efficiency. Stage 3. The RNA world, life's first information system and catalyst. The chicken or egg dilemma of DNA and proteins led to the insightful RNA world hypothesis, first proposed by Carl Woese, Francis Crick, and Leslie Orgel in the 1960s. The central idea is that RNA molecules could have performed both the genetic information storage and catalytic enzymatic functions in early life. This duality makes RNA a powerful candidate for the precursor to modern DNA protein life. Let's unpack the remarkable versatility of RNA. Information storage. Like DNA, RNA is a linear polymer of nucleotides. The sequence of these nucleotides can encode genetic information. RNA is generally single-stranded, but it can fold into complex three-dimensional structures, allowing it to perform functions beyond simple information storage. Catalysis, ribozymes. This is RNA's secret weapon. In the 1980s, Sidney Altman and Thomas Scheck independently discovered that certain RNA molecules could act as enzymes, catalyzing specific biochemical reactions. They called these catalytic RNAs ribozymes. The discovery of ribozymes was a monumental breakthrough, providing empirical support for the RNA world. Ribozymes can cleave and ligate RNA strands, synthesize peptide bonds, and even catalyze their own replication. In an RNA world scenario, early life might have consisted of self-replicating ribozymes. Imagine a pool of activated RNA nucleotides. A particular ribozyme, by chance, folds into a shape that allows it to bind to other nucleotides and catalyze their linkage into a complementary RNA strand. If this new strand can then act as a template for another copy, the system has achieved self-replication. Errors in this copying process would introduce mutations, creating variations upon which natural selection could act. Ribozymes that were more efficient at self-replication, or more stable, or better at catalyzing other useful reactions, like synthesizing precursor molecules, would proliferate. This would be the very beginning of molecular evolution. Evidence for the RNA world hypothesis comes from several lines of inquiry. Discovery of ribozymes. As mentioned, the existence of naturally occurring ribozymes in modern cells provides strong support. The most famous example is the ribosome, the cellular machine that synthesizes proteins. The catalytic core of the ribosome, the part that actually forms the peptide bonds between amino acids, is made of ribosomal RNA, rRNA, not protein. This suggests that RNA was the primordial catalyst for protein synthesis, a function it still performs today. RNA in fundamental processes. RNA plays crucial roles in many other fundamental cellular processes today, beyond just protein synthesis. It's involved in gene regulation, e.g., microRNAs, splicing, snRNAs, and viral genomes. Many viruses use RNA as their genetic material. These widespread and ancient roles suggest RNA's deep evolutionary history. Simpler prebiotic synthesis. While synthesizing RNA nucleotides is challenging, researchers have made significant progress in demonstrating plausible prebiotic pathways. The foremost reaction can produce sugars from formaldehyde, and various pathways can lead to nucleobases. A key challenge has been demonstrating the simultaneous and efficient formation of all the components and their linkage into a functional nucleotide under single, plausible early Earth conditions. However, innovative experiments, such as Sutherland's work mentioned earlier, are demonstrating ways these components could have arisen simultaneously. Greater stability of DNA. DNA is chemically more stable than RNA. The deoxyribose sugar in DNA lacks a hydroxyl group that is present in ribose, making DNA less susceptible to hydrolysis. Additionally, DNA uses thymine instead of uracil. The deamination of cytosine produces uracil, and if DNA used uracil, repair mechanisms would struggle to distinguish between normal uracil and deaminated cytosine. The use of thymine allows for efficient detection and repair of deaminated cytosine, protecting the integrity of the genetic code. This suggests that DNA evolved later as a more robust and stable repository for genetic information, taking over from the more reactive RNA. Despite its compelling nature, the RNA world hypothesis still has open questions. The spontaneous formation of long, complex RNA polymers under early Earth conditions remains a formidable challenge. The fidelity of abiotic replication might have been low, leading to error catastrophe. And the origin of the first activated nucleotides themselves, molecules with enough energy to polymerize, is an area of active research. Some scientists propose a pre-RNA world where simpler, more easily formed nucleic acid analogs, like PNA or TNA, served as the initial genetic material, later giving way to RNA. Stage 4. Compartmentalization, the birth of protocells. A self-replicating system, however sophisticated, is not truly alive in the way we understand it, if its components are simply diffusing freely in an open environment. Life requires boundaries. It needs to create and maintain an internal environment distinct from the external one, to concentrate its molecules, and to protect its delicate machinery from degradation. This is where the cell membrane and the concept of a protocell becomes indispensable. Modern cell membranes are sophisticated structures primarily composed of a phospholipid bilayer with embedded proteins. These membranes are selectively permeable, controlling what enters and exits the cell, and are crucial for maintaining cellular homeostasis. The spontaneous formation of such complex membranes abiotically is unlikely. However, simpler lipid molecules can self-assemble into basic membrane structures. Fatty acids, which are simpler precursors to phospholipids, are known to form spontaneously under plausible early Earth conditions, e.g., from the breakdown of waxes or delivered by meteorites. When these fatty acids are introduced into water, they spontaneously arrange themselves into spherical structures called vesicles or micelles. This occurs because fatty acids are amphiphilic. They have a hydrophilic, water-loving head and a hydrophobic, water-fearing tail. In water, the hydrophobic tails cluster together to avoid water, while the hydrophilic heads face outwards, interacting with the water. This forms a stable bilayer sphere. These simple fatty acid vesicles possess several key properties that make them excellent candidates for protocells. Self-assembly. They form spontaneously, requiring no complex machinery. Encapsulation. They can trap molecules, including self-replicating RNA and catalysts, within their internal space. This concentration is vital for increasing reaction rates. Selective permeability. While not as sophisticated as modern membranes, they can still control the passage of some molecules, allowing small nutrients to enter while retaining larger encapsulated polymers. Growth and division. Perhaps most remarkably, These vesicles can grow by incorporating more lipids from their surroundings. If they become too large or are subjected to physical stress, like shear forces in flowing water, they can spontaneously divide into smaller daughter vesicles. This simple form of replication, coupled with the ability to encapsulate a self-replicating genetic system, allows for protocellular selection. Vesicles that are better at growing, dividing, and protecting their internal contents would have a selective advantage. Imagine a scenario where a self-replicating ribozyme system gets encapsulated within a growing and dividing fatty acid vesicle. This creates a proto-organism, a genetic system contained within a boundary, capable of interacting with its environment in a controlled way and subject to selection pressures. Errors in RNA replication or variations in vesicle composition could lead to different traits. For example, a protocell with a slightly more permeable membrane that could take up nutrients more efficiently, or one whose encapsulated ribozyme was better at synthesizing membrane components, would outcompete others. The interaction between the encapsulated genetic material and the membrane is critical. Some theories suggest that early RNA molecules might have even evolved to interact with or influence membrane properties, perhaps by synthesizing primitive membrane-stabilizing molecules or even simple transport channels. This feedback loop, where the genetic material influences its own container, further accelerates the path towards true cellular life. Stage 5. The transition to DNA and proteins. The last universal common ancestor, LUCA. The RNA world, while a powerful hypothesis, was a transitional phase. Modern life is built on a DNA protein system. How did this transition occur? The consensus is that DNA and proteins gradually took over the primary roles of information storage and catalysis, respectively, due to their superior properties. DNA for stability. As mentioned, DNA is more chemically stable than RNA. Its deoxyribose sugar is less reactive, and the presence of thymine instead of uracil provides a more robust system for error correction and maintaining genetic integrity. In an increasingly complex world with larger genomes, a stable, long-term archive of genetic information became essential. The evolution of enzymes, proteins, that could synthesize DNA from RNA templates, reverse transcriptase-like activity, and then replicate DNA polymerase would have been crucial steps in this transition. Proteins for versatility. While RNA can catalyze reactions, proteins offer a much greater range of catalytic power and structural diversity. Proteins are made from 20 different amino acids, each with unique chemical properties: acidic, basic, polar, nonpolar. This allows proteins to fold into incredibly diverse and precise three-dimensional shapes, enabling them to catalyze an enormous variety of biochemical reactions with high specificity and efficiency. As the complexity of nascent life increased, the demand for more varied and efficient catalysts would have driven the selection for protein-based enzymes. The transition from an RNA world to a DNA protein world would have been gradual, likely involving a period where all three types of molecules coexisted and interacted in increasingly sophisticated ways. RNA would have initially directed the synthesis of primitive proteins, and some of these proteins would have been enzymes that improved RNA replication, while others might have helped in the synthesis of DNA precursors. Eventually, a highly efficient system emerged where DNA stored the instructions, RNA acted as an intermediary and regulator, and proteins performed the vast majority of cellular work. This transition period ultimately led to the last universal common ancestor, LUCA. LUCA is not the first life, but rather the most recent common ancestor of all extant life on Earth: bacteria, archaea, and eukaryotes. While we don't have a fossil of LUCA, scientists infer its characteristics by comparing the universal features shared by all life forms. LUCA likely had a lipid membrane, a genetic code based on DNA, though perhaps some RNA elements still played a central role. Ribosomes for protein synthesis, using rRNA as the catalytic core, a basic set of metabolic pathways to extract energy and synthesize cellular components. The ability to divide and reproduce. Luca lived approximately 3.5 to 3.8 billion years ago. The evidence for Luca comes from the striking universality of the genetic code, the shared machinery of protein synthesis, and highly conserved metabolic pathways across all domains of life. This universality points to a single common origin for all life forms existing today. Remaining questions and future directions. While the framework of abiogenesis, from prebiotic chemistry to protocells and the RNA world, is well supported by experimental evidence and theoretical considerations, many significant questions remain open. The when and where. While we have plausible scenarios, hydrothermal vents, wet-dry cycles on land, pinpointing the exact conditions and location where life first emerged remains elusive. The early Earth was a heterogeneous planet, and life could have arisen in multiple places, with one lineage ultimately dominating. The problem of homochirality. How did early life consistently select for only L-amino acids and D sugars from a racemic mixture? While some mechanisms have been proposed, a definitive universally accepted explanation is still lacking. The origin of activated monomers. How were the nucleotides and amino acids sufficiently activated with energy-rich phosphate bonds to drive polymerization in the absence of ATP? While some prebiotic phosphorylation reactions have been demonstrated, a complete pathway for the efficient and sustained supply of activated monomers is still under investigation. The origin of the genetic code. The genetic code, which translates nucleotide sequences into amino acid sequences, is nearly universal. How did this specific, seemingly arbitrary mapping between codons and amino acids first evolve? Was it a frozen accident, or were there chemical affinities between early amino acids and their cognate RNA sequences? From protocell to true cell, the transition from a simple fatty acid vesicle with encapsulated RNA to a complex, regulated cell with integrated metabolic pathways, protein synthesis, and DNA replication is a vast evolutionary journey that is still being explored. The metabolism first versus genetics first debate. While the RNA world hypothesis emphasizes genetic information, some researchers argue for a metabolism first approach. They propose that self-sustaining chemical reaction networks, perhaps centered around mineral catalysts at hydrothermal vents, emerged first, creating an environment that could then support the later development of informational molecules. Both views have strong proponents, and it's possible that elements of both were intertwined in the earliest stages. Future research in abiogenesis is highly interdisciplinary, drawing on chemistry, biology, geology, astronomy, and physics. Scientists are conducting more sophisticated laboratory experiments to simulate early Earth conditions, using a wider range of atmospheric compositions, mineral catalysts, and energy sources. Searching for more extraterrestrial organic matter in meteorites and comets, and planning missions to ocean worlds like Europa and Enceladus, which might harbor active hydrothermal systems potentially conducive to abiogenesis. Developing computational models to simulate the chemical networks and evolutionary dynamics of early life. Exploring alternative biochemistries. Could life arise with different genetic polymers, different solvents, or different elemental compositions? This helps us understand the fundamental principles underlying life's emergence. The study of abiogenesis is not about definitively proving a single narrative, but about building a robust framework of plausible pathways, supported by empirical evidence. Each new discovery refines our understanding, pushing back the curtain on one of the deepest mysteries of existence. The journey from sterile rock to vibrant biosphere is a testament to the inherent creativity of matter. the relentless drive of energy, and the surprising capacity of chemical reactions to self-organize into systems of breathtaking complexity. It reminds us that life, far from being an anomaly, might be a natural cosmic phenomenon, waiting to ignite wherever the conditions are just right across the vast expanse of the universe. The impossible spark, it turns out, might be a fundamental property of the cosmos itself,